Hi, I'm Mary Beth Horsington on behalf of the Strathmore Speaker Series. Thanks so much for stopping in here tonight for our first event of the 2021 season. Under normal circumstances, we'd be holding this event at the Fire Barn in Onondaga Park in Strathmore, in the Strathmore neighborhood, but COVID has necessitated the switch to Zoom. Fortunately, the Onondaga Free Library is providing the technical assistance we need to bring you this program, and we're indebted to them for their expertise and their generosity. The Strathmore Speaker Series is made possible through the support of the City of Syracuse, the Gifford Foundation, and the Greater Strathmore Neighborhood Association. Our guest speaker tonight is Dr. Hyatt Green, a professor at the SUNY College of Environmental Science and Forestry. Dr. Green has developed a truly novel approach to battling COVID-19. It involves conducting wastewater monitoring and using the results as an early warning system for the next major outbreak of the virus in Onondaga County. Dr. Green holds a PhD in microbiology from Oregon State University, and his work has been featured in a variety of scientific journals. He has done extensive research in molecular, microbial ecology, water quality, and microbial source tracking. His project is a coordinated effort involving microbiologists, public health officials, and operational technologists working together to contain COVID-19 before it spreads. When Dr. Green completes his talk, there will be a question and answer session, so stay tuned to your Zoom screen. And now, please welcome our speaker, Dr. Hyatt Green.
Thanks, Scott. Thanks, Mary Beth. Let me just make sure we can get the presentation there. We have a thumbs up. You can see that?
Perfect.
Good. Thanks. It's an honor and a pleasure to be invited tonight for the Strathmore Speaker Series with the Onondaga Free Library. You know, we mentioned COVID fatigue just a minute ago. Sometimes I wonder if, you know, talking about this is, if we've almost had enough of it, right? Because unfortunately, you know, I think all of us have been impacted by this to some degree. And if it's all right with everyone, I will skip the, you know, a lot of the background because we are really familiar with the background, and I will skip a lot of the exponentially increasing plots that can be depressing sometimes. So and hopefully talk a little bit more about a pragmatic approach to fighting COVID-19 pandemic. So let's see. I think by now most of us have had some sort of COVID-19 test, whether it's from nasopharyngeal swab or a saliva swab or just spitting saliva into a tube like we do on campus sometimes for the tests that students get. This is an essential tool. This is absolutely essential for identifying the exact individuals that have COVID-19. It's essential for contact tracing. So that is one tool that we have to bring to bear. There are a number of limitations with this, and I think a number of these limitations were exacerbated early on in the pandemic when we were kind of learning how to fight this. The nasopharyngeal swabs or NP swabs or the saliva swabs, they often require a visit to the clinic. Not always, because those tests have now spread out as time has gone on, has spread out on campus and workplaces and things like that. But they often require a clinic visit, which means that a lot of the people that actually have the infection are not showing up, and they are invisible infections. And you've heard this term before, asymptomatic carriers, people that can spread the disease, but that don't know they have it, right? Another thing that we had issue with, this is more so early on, I think the turnaround time has improved, but it could be a 5 to 7 day average turnaround time for these individual tests. And I've heard, I think I've had neighbors have 10-day turnarounds. And so you wonder how useful is a positive test after it's been 10 days after the sample was taken. It's either impossible or just totally impractical to test everyone or even large portions of the population. That just takes too many resources on administering the test, on collecting that swab, but also testing that swab. It's just not feasible. So we took an approach of this gives us the idea that, well, maybe we can look at wastewater, that maybe we can find this virus in wastewater in upstate New York, as was done elsewhere, and quantify it. And that will allow us to gauge the level of infection over a large portion of the population. The downside of such an approach is that it doesn't identify the infected individual, so you can't contact trace, right? But then again, that's not really the point, right? The idea is to get a basic idea of the level of infection. And one of the first bits of data that came out that suggested that we could actually do this was this the data on the right here that I'm showing, and basically looking at the level of SARS-CoV-2, which is the virus that causes COVID-19, in stool or feces in compared to sputum, right? And sputum is this reminds me to give you a late warning that we will be talking about some unsavory substances throughout this talk. So sputum is basically mucus and saliva mixed together that's coughed out of lungs. It usually has a large amount of virus, and shown on this plot has that stool has pretty much an equal amount throughout the course of an infection. And so we know that there's lots of virus being shed and flushed. And a single sample of wastewater is going to represent hundreds or even thousands or tens of thousands of people, depending on where that sample's taken, the size of the city, the size of the sewer shed. And there's some advantages to that. It's de-identified and it's non-invasive. So there's this aspect of privacy that's no longer a concern, but also it doesn't require someone to show up to a clinic or to actually walk over and get a test. It is so by that we're detecting individuals that don't know they're infected, in addition to those individuals that are symptomatic. And we can do this a lot faster, it turns out, right? So we're right now we're doing this at about, and I'll give you a brief idea of how we do this, but it's very efficient. We can get about a 48-hour turnaround on this type of analysis. And so here's the basic idea. We have a certain portion of the population that are reported infections, but we have a lot more individuals in that population that are unreported, but they are still shedding the virus, and they are still important epidemiologically because they can spread the disease. This gets into the sewer, goes into the treatment plant. We typically take samples, not always, but at or near treatment plants. This will be before the wastewater is treated, because the treatment process, thank goodness, it is very effective at destroying this virus and most other pathogens. We take that sample, we measure the amount of viral RNA, basically its genome, in that wastewater sample. We do some data analysis to get an idea of the trend over time, whether it's going up or down. And then we use that to adjust COVID-19 control measures. At least that's the idea. Whether we should, remind people that, hey, virus is spreading in your area, make sure you social distance, these public service announcements, or more drastic measures. And this was one of the first bits of data that I saw that said, wow, this could actually work. And this was a very inspirational paper for me from Paris by Wurtzer and colleagues. And I'll take you through this. In gray, in kind of the light gray, you have hospitalizations from COVID-19. And I believe here in dark gray, you have deaths. Or excuse me, these are cases in light gray. And so up on the top panel, you have the wastewater concentrations of SARS-CoV-2. And you can see that the trend over time sort of follows that of the cases in Paris. And what gets me about this, you see this first sampling date here and how there were just a few number of cases in a relatively large city. And this was the first detection, this was the first detection because it was just the first time they sampled, right? I always wonder, wow, if they had started sampling earlier, a few days before or a few weeks before, would they have caught the virus? Would they have observed the virus within the city limits before there was a single case, right? And so that led us to think, well, maybe that wastewater surveillance has a predictive capacity or has an early warning capacity that could tell us where the virus has gotten into, but an official case or a positive test hasn't shown up yet. So our research questions started from the what we thought were fairly easy to answer, working up to more complex and more difficult to answer. One, can we detect or quantify SARS-CoV-2, with quantification being a lot better in upstate New York wastewater, like was done in the Netherlands and Paris, and I think in Boston. So I think those were the three places that it had been done by the time we really started our work. So we kind of knew that was kind of a safe bet, that if we were doing things right, we could probably at least detect it, if not quantify it in upstate New York, right? Because we don't think upstate New York is that different epidemiologically than these other places. If we could detect it, does it serve as an early warning of future outbreaks? And I say near future there because, right, we're not going to detect outbreaks months in advance, right? That's unrealistic. I'm talking about a few days to a week, maybe. If we can quantify it, if we can put a number, right? If we can say there's X amount of this virus in wastewater, does it have a relationship to infection rates on the ground, right? You know, does it relate to these classic epidemiological parameters that we see, like deaths, hospitalizations, positive cases, or cases, things like that? Because if it doesn't, then then we're just having fun, right? If we're not relating it to something epidemiologically important, then it doesn't really matter. And then finally, and I think the most ambitious kind of research question that we wanted to answer was, can wastewater surveillance forecast the number of infections in the near future, right? And you can see the obvious benefit to that. If you can say 3 days from now, we're going to have X number of new cases, that allows public health teams to make whatever the adjustments are that they need to do. It allows them to prepare. So those were our research questions. And so at this point, I'll just give you an idea of how fast things progressed. And I know the font there's a little small, but just to give you an idea of the timeline on this. So this was right before spring break when all this kind of started, at least spring break for ESF, and we went remote, I think right after spring break. And so At the same time, all the research labs were shut down. Unfortunately, right around that time is when I got an email from Dave Larson, who is a SU professor of epidemiology, who, I'm the lab guy, I'm the methods guy. Dave is really the leader of this whole effort, really coordinated us to put this together. And then we got in the lab 2-3 weeks later. Two of my students, Max Wilder and Ari Finty, jumped in the lab. I think they deserve a tremendous amount of credit for right when cases are increasing the highest in Onondaga County. They came out of isolation, came in the lab and worked to develop these methods. We then, about a week later, we developed the sucrose cushion method. And a week later, after that, we had that first detection in Onondaga County wastewater. And I believe, thinking back months ago, this was probably the second area in the U.S. where SARS-CoV-2 was detected in wastewater outside of the Boston area. And then 2 weeks later, we published it. We thought we found a pretty nifty way to do this. And so we wanted everybody to know about it. And so we published it on a preprint server, free, available, and we let everybody know how to do what we did, so that if they found it useful, they could use it in their area. Okay. So we got back to the lab, and this was, this is a tough situation, I think, for my students. This is Max, processing some wastewater here. And this is a pretty unusual situation where usually you don't have a camera pointing at you when you're doing these methods. And so a lot of credit goes to them for working, on this stuff to develop these methods with a lot of scrutiny and a lot of cameras facing their way. So, and a lot of credit also goes to Frank Middleton at Upstate University. So you might have heard him if you keep up with Syracuse news. He's been all over Syracuse.com for developing the saliva testing and pooled saliva testing that has really helped, keep track of infections. And so this is actually a lab at Upstate Medical University. So we had to figure out how to do this. This was not like research in normal times. We had to have a method that was sensitive. Obviously, if you can't detect a virus, then it doesn't matter. It has to be affordable. The idea from the very beginning was to make this, if we can figure out how to get this to work, then everybody's going to use this, right? We're going to have to analyze so many samples and if it's too expensive, then that's no use to us. So we wanted it to be affordable. We wanted it to be fast, because if it takes as long as the swab testing used to, 5 to 7 days, then you lose predictive capacity, right? You lose that early warning. And it also had to be scalable, kind of along the idea of affordable, right? We had to be able to analyze hundreds of samples a day or thousands of samples a week to be useful, for public health officials. There were other methods to do this at the time, but tangential flow, like I'm kind of showing some of the equipment here in the first image, it was too slow. We can analyze one sample every 3 hours. It was really sensitive, but it was just way too slow. PEG precipitation, which is widely used, was too expensive. And the method that was used in the Netherlands that we tried to do, we couldn't get a hold of the supplies. So we ordered these filters that are shown there on the right that were used to process the wastewater. We ordered them on May 1st, and we eventually got them on August 11th, right? So, because once that method was published, everybody just bought them up. And so this is really unlike research in normal times where you have the ability to plan and things like that. We really had to develop something new because we didn't have any other option if we wanted to do this, right? So I'll just give you a quick overview of the method and what's going on. So this is called the Sucrose cushion method. And it's really simple. It just requires a fairly expensive piece of equipment, right? And we were lucky enough to have an ultracentrifuge, so that wasn't a problem. All we do is we take a tube and we first put in 20 milliliters of wastewater. And then very carefully, we take a thin tube and we put a sucrose solution underneath that wastewater. And this is no different than the sucrose. This is table sugar, right? We add a couple other salts to it, you know, buffers and things like that, to change the chemistry a little bit. But we very carefully layer the sucrose cushion underneath the wastewater, right? So you get this clear cushion interface there. And then after we get about 6 of those tubes ready, we put them in an ultracentrifuge. And an ultracentrifuge, all it does is just spins things around, like a merry-go-round or a salad spinner, whatever you want to, whatever you want to use as an example of that. And it spins and applies a huge amount of force. So we're spinning things that results in a force of 150,000 times the force of gravity. And what happens, because the sucrose cushion is so dense, right? It's almost like a syrup kind of consistency. It's very dense. And so what happens is that allows a lot of our RNA, our nucleic acids that we're looking for, but a lot of other things to sink down to the bottom of the tube. But what also happens is that a lot of these low density contaminants, like undegraded bits of toilet paper, I mean, we're not exactly sure, right? But they're low density and they rest right on top of that, thereby separating out these contaminants that we don't want from the nucleic acids that we do want. Okay. And so that used to be, we've now scaled that up from 20 milliliters of wastewater to 40 milliliters of wastewater. So even though we've doubled it, it still doesn't take a whole lot of wastewater to, we're not working with a really harmful amount of wastewater in the lab. Working with small amounts is usually a lot safer. And so this is basically the overview of the method, how the workflow is going at the very beginning, where it was really a true collaboration between Frank Middleton and I at Upstate Medical University. And then we would take these pellets and bring them over to ESF and do the rest. So I'm not going to go into detail on this. Obviously, if there's questions on this later, I'd be happy to answer them. But you essentially, if you've gotten a swab test or a saliva test, then you might be familiar with the latter half of this process, because it's exactly the same, right? The only thing that's different is that we're processing the methods a little bit differently before that test is run, before that PCR-based test is run on the sample. So you can see, early on, we weren't quite sure how much material we needed, how much wastewater we needed. So we were getting liters and liters delivered to us. It kind of got to be a problem. Now we know we can work with a lot less, but you can see how dirty this wastewater is, how much is in it, and I'll be honest, if someone said we were going to be successful at getting RNA out of wastewater, when we started this, I would have thought they were crazy, but it really works. So this is just to give you an idea of the sampling. We don't have someone standing there, by the wastewater stream collecting a sample every once in a while. We actually use a machine to do that, and that's nice because we can program these machines to collect a sample every 6 hours. Right, and then so for every 24 hours, you collect 4 subsamples. All of those 4 subsamples are then combined into one bottle, and then we take that. So you can see this is a ISCO automated sampler, and a tube sticking out of it going into the manhole. And at the end of this tube, you can barely make out there like a weight, like a heavy hollow tube of metal has been, taped to the end of that. That's just a weight so that the tube rests in the wastewater stream so that we're not drawing up air, while we're trying to pull out wastewater. So all of those subsamples, will be combined in this one, what looks to be maybe a 5-liter container. And, this was then a technician can go out and once a day and collect these large samples, shake it, and then basically put it into what is basically a well-labeled milk jug. And so it's actually quite a lot of work to go around and collect all these samples. But this sample here, when we get that, you think about that sample has been in the wastewater system for at least hours, right? Sometimes days, right? And so our question is, well, what's happening to that virus while it's in the wastewater system, right? There are actually a number of factors that can limit our detection of the virus in wastewater. And one of the key questions that we have, remember, one of the key research questions is are the concentrations of SARS-CoV-2 in the wastewater, can they be related to the number of infections? That would be that's the end goal, right? But what's limiting that are a number of factors, including dilution. So every time you take a shower, right, that's adding in a lot of gray water that dilutes down the exact virus that we're looking for. Every time it rains, I mean, we're in an area where our wastewater infrastructure is not so new, it's pretty old. And so there's lots of cracks in our wastewater infrastructure where stormwater can get in there, wastewater can get out. Well, actually, we have a combined system, right? So stormwater is actually intentionally combined with wastewater. So stormwater will dilute down our virus. You have some industries that will release a lot of water or other things, right? That also serve to dilute our virus. And you have infiltration, inflow. These are just, where water makes its way into the wastewater system where it's not supposed to be. Then you have the virus falling apart. So luckily, I think it's a good thing in the end that this virus is really weak. It's really not resilient to environmental factors. That does make our job harder, but I think in the end, it's a great thing. So how does temperature affect the decay of the virus? What about pH? And then we also have those same industries that are releasing certain compounds that are useful for manufacturing processes that interfere, that might break down that RNA that we're looking for. So one way to approach this problem is to put a bunch of sensors out in the wastewater infrastructure and then kind of use a bunch of statistics to estimate for these factors. That's really hard and really expensive. So our question was, can we find a surrogate that behaves a lot like SARS-CoV-2 in the wastewater during its transit, right from the flush to when we take a sample? And when I say surrogate, I think of something that is accumulating these effects of decay and dilution in the same way. It's like a stand-in for SARS-CoV-2, right? And so a surrogate is going to be really, it's going to be in our microbiomes. It's going to be at really high concentrations. And this is one of those times where, I don't know, I guess I feel very lucky to have the training that I have. And I'm just to take a step back and a step to the side real quick. I think a lot of us, most of us, were not obviously studying coronaviruses before this happened. And so, we had to shift everything that we're doing to focus on COVID-19 so that we could get out of this pandemic faster. So I came from a field called microbial source tracking, and a central task of microbial source tracking is to identify microorganisms that are very abundant in the gut, but also very widespread. And the central goal is to estimate the types and the quantities of different fecal sources. Typically, this is in ambient water samples. And so through my training at Oregon State University, but also at the US EPA, we developed plenty of these methods. And fortunately, we one of those methods was turned into an official validated EPA method, which it doesn't seem like a big deal, but when you think about that, that the whole world really follows what the EPA does. This means that our method's now being used worldwide to estimate fecal pollution in typically ambient water samples. And so just to show you a little bit of how that data is used. So this is a study, I think right when I arrived at ESF, I think the very next summer in 2015, we went out, me and an undergraduate student went out and sampled all these places over Onondaga Creek. This was also a collaboration with Onondaga Environmental Institute. And we ran these microbial source tracking assays on ambient water samples on Onondaga Creek with the main goal of trying to identify what were the major sources of contamination in Onondaga Creek. And so in Terra 1, that's just a general source, that's a general measure of fecal contamination. It's not source specific. And you can kind of see that on this one sampling date, we had a lot in the urban area, right around downtown Syracuse. That's no surprise, right? We have, like I said, a very leaky wastewater infrastructure. And so that was no surprise. Also not a surprise, when we ran human markers, we see a lot of human contamination also around, downtown Syracuse and further south around the urban areas. And then rum-2-bac, that's actually a test for ruminant contamination or cow fecal contamination. Typically, this correlates with agricultural areas. And so that's exactly what we see here, is in more of the agricultural areas, we see sources of ruminant contamination, right? And so none of this is a real surprise, but this is the difference between having an inclination that contaminants are coming from a certain source versus evidence. And so that is typically how these methods are used. But we wanted to kind of rely on this field of microbial source tracking to find an appropriate surrogate for SARS-CoV-2 in the wastewater system. And so the one that we went to, which has a pretty hilarious name considering its exit strategy is through the rectum, is called crassphage. And yes, the A is capitalized. I don't know who named this, but I'd really like to have a conversation with them. It's really named, it's really cross assembly phage is the full name. But of course we say crassphage. Anyway, so this organism has been studied for, I think, since 2014. So for, well, maybe earlier than that, maybe 2012. So for about 8 years. So we know a lot about crassphage. We know that it's cosmopolitan. It's found in about a third of us, depending on what we eat. And it's really abundant. So it's present at about 10 to the 7th. That's about 10 million to 100 million viruses per gram of fecal material. It's really well studied, and the detection methods were available. Like we could just take this method. It was developed at the EPA. We could take this method and put it into use. And for our purposes, something that was fast was great. So, so we chose crassphage to act as sort of a denominator for the total amount of contamination that we're testing in wastewater samples. And so here's how that's sort of that's a schematic of how that's working. So we have a human cell, SARS-CoV-2, of course, infects that cell, and human cells are pretty weak when they get into the wastewater infrastructure, and so they break immediately, they lyse immediately, and release their genetic material, including that viral material. Crassphage, it does the same thing, except for a bacteria as the host. So, excuse me, and we can take those, we can co-monitor both of those things. Both of those will experience decay and dilution. And then in a sample, we can measure both of those. And then we can sort of use crassphage as a denominator and SARS-CoV-2 as a numerator. And this should give us an idea of the relative levels of infection within the community, accounting for dilution, accounting for decay and things like that. So we know that crassphage is proportional to the amount of crassphage that we find in wastewater is proportional to the amount of people that are represented per unit flow, right? So the more concentrated the material is, the higher the abundance of crassphage. And so that's a really good sign that, hey, crassphage it's behaving the way that we thought that it would. And this is data from Onondaga County. And so that's just confirmation that, hey, the more people that are represented, the higher the amount of crassphage. So that's more evidence that it would be a good denominator. So these are some of the first results that we published in late May. And in the left panel is cases. And so these lines, these kind of gray lines here, these darker gray lines, those are the sewer sheds or service areas. Unfortunately, we don't collect epidemiological data based on sewer sheds. We collect it based on zip codes. So these blotches of color, these are zip codes. And so you kind of have to imagine the overlap there. But what we saw was when we take the SARS-CoV-2 to crassphage ratio, we saw that the urban area where you have the most people and the most infections, the highest rates of infections, we saw the highest signal of crassphage in that same area. And so that was kind of the first indication in Onondaga County, that we saw this spatial correlation between SARS-CoV-2 in wastewater and the epidemiological parameters that were also being measured, test results, basically. I wanted to show you some more. So I showed you some old data from May. I also wanted to show you some more recent stuff, some recent results. So Bruce Adams from Cortland Wastewater Treatment Plant gave me this data this morning and gave me permission to kind of talk about it. But basically, just to orient you in the kind of pink squiggly line, those are new daily cases. So the amount of new cases that were observed in Cortland County every day. And you can see how much variance there is to that number. And you can also see that's why we have to smooth it to account for that variance. Oops. And so you see these humps, these peaks in these cases. And if you look at the wastewater data, if you look at these light blue points, those are the individual wastewater numbers. when we take SARS-CoV-2 over crass phage, they're all over the place, right? And it kind of, maybe you can make out a trend, but it's not until you add a trend line till you see that the wastewater numbers reflect really well the number of new cases per day in Cortland. When this peak goes up, you see maybe a little bit beforehand, you see also a peak in the wastewater numbers. You see it go down maybe a week beforehand, you see that same downward trend in the wastewater. And then you also see a peak over more recently, maybe 3 weeks ago, at least looking at the trend line. And then a few, maybe a week before that, you see a peak in the wastewater. We can do more analyses on these numbers, and basically, we can tell that a 0.25 increase in this ratio is approximately, now we're working to decrease some of the variance there, but a 0.25 increase in that ratio is approximately equal to about a 10 times increase in the number of active cases a few days to a week in advance, right? So, this gives us some early warning. I mean, if you think about this trough here, when it started to go up, well, the wastewater data started to go up, it's hard to tell, you'd have to look at it more closely, but maybe a week or so beforehand. And so you think about the kind of warning, this kind of valuable warning that this would give to public health officials to take the appropriate measures, whatever they thought was appropriate. So our methods, at least incorporating crass phage, have been reproduced in the Netherlands and other places. So this is a similar plot in Amsterdam. And in blue, these are the cases. This is a moving average. The blue line is a moving average of cases over time. The gray line is hospitalizations. And then the orange line there is their SARS-CoV-2 over the crass phage, SARS-CoV-2 crass phage ratio. And you see that it tracks, I think pretty well in Amsterdam as well. So it's not just Cortland, it's also bigger cities like Amsterdam too. So, and I was particularly happy to see this because this was the group, KWR was the group that the first group in the world to demonstrate wastewater surveillance of SARS-CoV-2. And so to see that group pick up our crass phage method, I was pretty happy that they thought that would work for them as well. If you remember back to fall, if you remember back to summer, actually, we were all, I think we were all freaking out, at least as professors, we're all freaking out for like months. What are we going to do in the fall when all the students come back, right? And, we were all very worried. And I mean, ESF, and I would say SU did remarkably well. I can't remember, ESF had like 3 cases in the fall. I mean, we have a small student body, but we had a really small number of cases. So I think we did really well. There were some other schools where we were measuring wastewater that did not do so well. So SUNY Oneonta on the left plots there. So we have cases on top, and then the wastewater data on the bottom. SUNY Cortland, we had a little outbreak, which was also detected in wastewater. And then SUNY Oswego had an outbreak as well, which also showed up in the wastewater. So I want to zoom in onto SUNY Oneonta real quick, because I think that was one of the most ferocious outbreaks that we heard a lot about, how it just exploded on campus. And so we scrutinized those results. There we go. So this is kind of a zoom in on that left panel that I was showing you earlier. And So we had wastewater surveillance begin on August 4th. We had one background detection. You know, it was below the limits of quantification, but it was still there. Students returned to campus August 17th, and not long after, probably after a few parties, on August 20th, we had another low-level detection. And then on August 24th, we saw the first quantifiable SARS-CoV, first quantifiable wastewater sample. So it's at a higher level than we've seen thus far in this area. And then the next day, we have the first positive COVID-19 test. And so in this case, we're only getting 1 day warning, which is better than nothing. But I always think about, well, man, what if we were monitoring every day? I mean, the wastewater monitoring is really cheap. Why didn't we just, in retrospect, why didn't we just monitor every day, twice a day? We would have had a much better warning and potentially could have prevented or slowed down an outbreak, right? So things got so bad, they initiated a campus pause. And then in the meantime, levels kept going up, kept rising along with cases. And then students, they said, we've had enough of this. So students left campus, or they began to leave campus. Importantly, so you might be asking, well, why do wastewater levels keep going up? Why do they keep going up after students have started to leave, right? It should go down because, well, that's because a lot of the students that were infected were quarantined, and we didn't want them going back home infecting their friends and family, right? So they were still there contributing SARS-CoV-2 and crass phage. And so once those students got through with quarantine, then we see the drop off. Then we see the numbers start to go down, and because they started to leave. So back to the timeline, this got too big for our britches, really. It was me and two students, right? And a PhD student, undergraduate student, and we thought we did a pretty good job, but we got overloaded with samples, and so we had to partner with we partnered with a private firm, private biotech company, Quadrant Biosciences, and they took over analysis because we could analyze maybe 30 samples a day. And so we saw that where we saw this going was like, well, we need thousands of samples a week. That's the capacity we need. And then eventually we worked with New York State to get a pilot study. So just to give you an idea of the capacity, we have a Quadrant, I should say, has 4 ultracentrifuges with a capacity for 2,000 samples a week, all with a 48-hour turnaround time. At least during the school year, they were measuring dorms at 13 SUNY schools, 10 different municipalities at least, although that number has changed since then, since I've presented this last. And I'm trying to remember when, probably around October sometime, we started measuring all nursing homes and assisted living facilities in Onondaga County. So we're getting quite a bit of data, right? But I always think, well, what good is that data if it's not making it to the people that matter, right? And great, I get to see the data, but I'm a microbiologist, right? So I can only take it so far. It's important that we thought it was most useful to the public health managers, wastewater facility managers, to have easy access to that. And so our collaborator, Mary Collins, also at ESF, developed, and her student, Dustin Hill, developed a dashboard, a data dashboard, so that we can see what is the alert level based on the rate of change. How fast is it going up, or how fast are these concentrations going down? But also a confidence in that, right? Sometimes the variance or the change is just too high to be confident in that increase or that decrease. So we're providing both a slope and a confidence in that. they can also click on, as long as we have the geographic info, the geospatial information, they can click on these individual sewer sheds and get an idea of what's the increase in the alert level and the confidence in these individual sewer sheds. And so this is available to, it's not widely publicly available, but it is available to wastewater municipalities and that are partners and public health officials. Okay, so what do we do now? Like, where do we go from here? You know, I've only shown you information on how to detect the original SARS-CoV-2 strain in wastewater and what we do with that, how it might be useful. What we could also do is strain determination from wastewater, so we can get an idea of what are there any new strains circulating in the population? Well, what proportion of the infections might those new strains be versus the old strain? We might be able to get an idea, as long as we can detect antibodies in wastewater, which we're also trying to do. Can we detect levels of immunity? Whether that's herd immunity or if that's immunity due to a vaccine, particularly important right now, as people are signing up for vaccine appointments and actually getting them. So hopefully we won't have to worry about COVID-19 to this degree for a long time. So wastewater could potentially be very useful for other pathogens, right? We know that we can detect, we've detected flu, influenza in wastewater as well. What about other viruses, adenovirus or norovirus or retroviruses? So there are a wide number of applications, I think, beyond COVID-19. So I'm going to wrap up here, but really, this was a collaboration unlike others I've been a part of. And it was with 3 universities, me and Dr. Collins at ESF, Dr. Larson's, Dr. Kamush, Dr. Zhang, Dr. McKnight at Syracuse, and Frank Middleton at Upstate Medical University. Really couldn't have been possible without all of us. You know, we'd also have to, you know, want to give significant credit to Quadrant, because without them, we wouldn't be able to scale up. We'd still be at that 30 sample per day, you know, limit. Arcadis was really useful in helping collect the samples, getting information about the service areas or sewer sheds. And the New York Department of Health, we got $500,000 from them to do a pilot in September and October. And all of this, of course, like we're all doing, we're doing right now, it was all over Zoom. So this is really interesting. A lot of these people I haven't met face to face, but I know them very well. I think that's about it. If we want to do a Q&A, Scott or Mary Beth, again, I really appreciate the opportunity. It's really a pleasure.
Well, thank you very much. Thank you very much, everybody there. And we have a few questions. Thank you, first off, to Hyatt for speaking at such length. And I think crassphage is now going to be everybody's favorite word to impress all their coworkers and relatives with this week. So we have a few questions from the chat. First question is from Kay Scott, and she asks, are you looking for the new strain or strains of SARS in the wastewater?
Yes, we are. Yeah, we haven't we're optimizing our methods now. So I don't have any data to show on that, unfortunately. We have a pretty good idea that it's here already. I know there was, let's see, there was some detections in Saratoga, I believe, and maybe elsewhere, but we know it's here. I would really like to get a gauge on how prevalent it is, right? I mean, because it's more infectious. So we would want to know, I mean, that there's a potential that might change our public health strategies, maybe a little bit. But I think by and large, I think still, you know, obviously wearing masks and all the precautions that we're used to taking, social distancing, things like that, are still helpful for that new strain. But we certainly want to know, right? We've seen how big of an impact that had on the UK and other places. There's another South African strain. There's a few of them now that are bouncing around. And so that is something that we are working on. But I don't have data on that yet.
Okay, the next question comes from Kathleen DeSantis. Sorry, Kathleen, hang on one second. Kathleen asks, is the aging century-old sewer water pipe system in Syracuse interfering with your data collection? I guess maybe aside from just that it's very leaky, but maybe the materials in the pipes erode into the water, that sort of thing.
Wow, that's a great question. Yeah, you know, we do sometimes, we notice, you know, little flecks of metal, right, or oxidized metal, black pieces of what seemed like basically black rust in our samples that may be interfering with our ability to detect, you know, the virus. Again, we have a control for that in crassphage, right? So we would think like if we have a hard time detecting crassphage, which is so abundant compared to SARS-CoV-2, you know, it's kind of a quality control measure as well. I didn't really focus on that, but crassphage kind of lets us know that we're doing things right and that we're not having that sample interference to a large degree. So that's a great question. I would imagine it is to a degree. But we haven't quantified that impact yet.
Okay, the next question is from Mary Beth, and she would like to know, did you literally drop everything else you were working on to focus on this project? And you know, we assume you were teaching and doing other research, and are you still focusing solely on this?
Yeah, it's a great question. Yeah, we dropped everything. And because I think, you know, I'm just so fortunate to have students that thought the same way that I did, that other research isn't going to move on. Our lives aren't going to move on until this is taken care of. And so if we can contribute in some way that can limit the number of deaths and hospitalizations and total illness, or even shorten the duration of the pandemic, even by 1 day, then it would be worth it. You know, I think we really have to realize, I think, how lucky we were to have expertise that was useful. And if it just seems like that was kind of, I considered it maybe a social duty, you know, that we need to do this. We need to set everything else aside and do this, do the best that we can, and expand it, and you know, increase the amount of data that is collected on this so that it is helpful. And you know, we weren't, you know, at the beginning, we weren't really sure how helpful it was going to be, but you know, we got to give it a shot. So.
Okay, the next question comes from Sam Hollister, and he asks, do you think the wastewater treatment plants around the country have the capacity to test and monitor their own effluents now and in the future, or would this still be something that a company
Yeah.
like Quadrant would have to do?
Yeah, that's a great question. There are some. There are some that can do it, and there are some that did it fairly early on. In the Southwest, I think because there's so much attention on water availability, there's a lot of funding and a lot of resources given to wastewater. Some aspects of like wastewater reclamation, they have a lot of resources. I think LA, I think Phoenix, and a few other places in the Southwest. They did have the capacity, and they bought the equipment to do this. They spent hundreds of thousands of dollars to gear up, and they started doing it. And that's great. Most places do not have the capacity and do not have the funding to do this sort of thing. I think the vast majority don't. And that was a limitation we ran into, is that, the money's got to come from somewhere to do, the resources have to come from somewhere to do this sort of thing. And obviously no one in 2019 budgeted for wastewater surveillance of a virus that didn't yet exist, right? So it had to come from somewhere, and those municipalities that did not have the resources just couldn't do it. They relied on academic labs or now service labs. So we did, we were pushing for federal funding and state funding of this sort of thing, but we found that it was to kind of support a broader effort. But we found that it was kind of difficult. I'm not exactly sure what the limitation, but my inclination was that it was difficult to convince epidemiologists, medical doctors that were trained in the clinical realm. It was hard. I think it's hard to get them to accept the utility of wastewater, right? And the fact that it's not identifying exactly who has the infection. We're still working on it, though.
Okay, next question is, you mentioned Los Angeles. Robert Bornhurst would like to know, is your process, aside from maybe Amsterdam that picked it up, are there other US cities, like for instance, Los Angeles, that are using your process right now?
Yeah, let's see. So there's a few down in Virginia. We have some collaborators that we're using this, and we're very well. And then a few campuses. There's a few independent labs. Let's see, Colgate and Clarkson. And I think those are the ones I know of. There are probably other places. There could be one I'm forgetting there, but there are other places. But I think they're really centered around, upstate New York. We weren't the first method on the scene, and so it's like one of those things. Even if the first method isn't the best, it's the one that gets out there and gets widely used. So, but our method is widely used in upstate New York for sure, or mostly upstate.
Next question is, are you seeing a downward trend now in Onondaga County?
Oh, good question. And you know what? I don't have an answer to that. I was spending most of the time looking at Cortland today. I have to ask Dave Larson. He keeps a better gauge on the epidemiological data. Shoot, I forgot to look at that. I'm sorry. Yeah.
All right, everybody will be hanging there. I saw a couple of hair, some hair raising in the thing. Everybody remain in suspense.
Oh, you know what? Here's why. I just remembered. There in Onondaga County, in the municipal wastewater samples, we are no longer receiving. So we are monitoring. We're monitoring wastewater, like I said, in nursing homes and SU when it's in session. And it's almost like we've gone. The analysis has gone upstream in the wastewater system to like individual facilities or groups of facilities. And so, unfortunately, Onondaga County was one of those that they had to make a tough decision on where to put their resources. And, the municipality itself couldn't fund wastewater analysis. So that's why. Sorry.
Well, that's okay. We'll all be, like I said, we'll all be in suspense for the next few days, waiting for
Yeah.
new numbers to come out.
Yeah.
Okay, the next question comes from Felipe Perez, and he asks, how much do you expect the introduction of the vaccine, or what effect, how much do you think that the vaccine will affect the data?
I hope it goes down, right? I mean, you would think that that it would lower infections, right? And so we would see those numbers go down. It's not like the old days where, some of the first vaccines were attenuated virus, right? Where they would heat up the virus a little bit. And so it would deactivate it, but the RNA might still be intact. That might contribute to, like, the vaccine itself might contribute to an elevated signal in the wastewater, maybe. But these vaccines, at least a couple of the first two that came out are mRNA vaccines, and that mRNA is translated and then degraded pretty rapidly. So I don't think that's going to be excreted. So we shouldn't see like a bump because of the vaccine. I think it should be a gradual decline that corresponds to a lower number of infections. It's a great question.
Okay, next question is, how much personal risk is involved in collecting and analyzing the samples?
Yeah. This is something that we had a lot of debate over early on, and my answer is no. It's no more dangerous than working with wastewater before the pandemic. This virus falls apart so quickly that by the time we get it, by the time we collect a wastewater sample, it's deactivated. It's not infectious. Now the RNA, its genome is still there, but it's the vast majority, if not all of it, is deactivated. So that being said, don't go drinking wastewater, because there's still plenty of other pathogens that are still plenty dangerous, right? I named a few of them earlier, right? Norovirus, adenoviruses, and other bacterial pathogens, Salmonella, Campylobacter. There's protozoan pathogens. I mean, there's a lot of potential there to get illness from wastewater without worrying about COVID-19 or SARS-CoV-2.
That's everybody's other takeaway. Don't drink wastewater. The library's public service announcement. The next question comes from Christian Bright, and she asks, when new strains or new pandemics do arise, do you have to receive a sample of the virus in order to recognize it? How does that initial setup for recognition of a specific virus work?
Oh, that's such a good question. I'll try to keep it brief. The reason, I'll start with the UK. The reason it's called, or it used to be called, we've changed the nomenclature since, but the reason it used to be called the UK strain is because it was first detected there, right? This new variant, this B117 variant. But it was detected there first, probably because the government funded a nationally funded genome sequencing program, not of wastewater, but of swabs. And so they have a huge bank of genome sequences. They were constantly collecting hundreds and hundreds of genome sequences a week, looking for variants and looking at the data. And so it's probably spread, it probably originated, or it's at least spread to other places. We just don't have as good an idea of it because we don't have that genome sequencing surveillance. And that's what we really need. We need that whole genome there. And once we have that published genome, this was like, you think back at the very beginning, the big turning point, not the very beginning, but the beginning of the pandemic, when we had that first Wuhan strain published online, right? And from that point on, we could develop a PCR-based technique which then we could go and develop nasal swab test. We could go and develop that PCR technique that's also used for wastewater, right? So that genome sequence, that closed genome is key, but that takes effort. That takes a lot of effort and resources, is to monitor for those new variants. But once that's done, we develop PCR primers. You can order PCR reagents for like $100 online, and they come within like 3 days, right? So once you have that genome sequence, you're ready to roll for pretty much any type of PCR-based detection in, I'd say, minimum 3 days.
Cool. All right, so we have 3 more questions, and I think we have time to fit them in. Patty Black, who is one of the members of the Strathmore Speakers Series Association, she asks, does this technology have other possible applications for other social indicators like opioids or maybe non-biological?
Right. Yeah, yes. And what we also have done, Dr. Tang Zhang at SU in the engineering department, he has taken subsamples of the same samples that we analyzed for COVID-19 or SARS-CoV-2 and analyzed for all sorts of things, all sorts of chemicals, opioids, but also opioid derivatives, and monitored, looked for the co-occurrence or maybe the correlation between these sometimes illicit drugs and COVID or SARS-CoV-2, excuse me. And there are some interesting, I don't know, there's some interesting correlations. One that sticks out to me is that pepper, it seems like there was more pepper. I think it's pepperine or pepperdine, which is a component of black pepper, and that increased along with SARS-CoV-2 infections. And although I don't know if that's the most important finding, that is the one that sticks out to me. And I always attributed that to people, cooking more at home or something and not going out to eat. I don't know. And using way too much pepper. I don't know. But I believe I'd have to look. That paper is published, and I'd have to look that up before I got you a good answer on the opioid question. Maybe he could be your next speaker. I don't know. He'd answer that way better than I would.
Okay, second to last question. Kathleen DeSantis asks, do you think this process will be able to identify future viruses and help us be prepared? Like, would we be able to detect something completely unknown?
Yeah.
Perhaps not, but something that would be odd.
Yeah. I get it. Yeah. That's something that that's something we're working towards, right? So, and I think we will be able to do that. The cost of sequencing, which is, I believe, what that task would rely on, is de novo pathogen discovery. Sequencing has gotten so cheap that some people, some I think fairly high-powered labs have been successful at taking a wastewater sample and just sequencing everything that's in it, and finding parts of SARS-CoV-2, the parts of SARS-CoV-2 genome. And so that would be challenging, but I think it's in the realm of possibility. I think we could probably do that as sequencing technology improves, as our knowledge of wastewater-based epidemiology as a tool improves. The challenge would be, when that was successful, there were lots of infections, and there was a lot of SARS-CoV-2 in the wastewater. So, I think the challenge would be, how do you detect something when, let's say it's something that's zoonotic, that hops from, a farm animal, a virus that starts infecting humans, right? Can you detect that first human, right, or this novel virus from that one person, in wastewater, and that's just not a whole lot of virus, and I think that might be the difficulty.
Okay.
But I think it's possible. I'm optimistic.
Got time for one more question, and it comes from Sandra Lipkowitz. And the question is, since you are a professor at SUNY ESF, are the SUNY schools, especially downstate on Long Island, where there's underwater wells and things like that, are they knowledgeable about your method? Is there communication within the SUNY system?
Yes, there is communication within the SUNY system, and I'd have to get a complete list of all the SUNY schools. I can't remember every one we're doing, but I think while school's out, we've taken a pause on that. Downstate, I do remember one SUNY school downstate, but I can't remember which one it was. I'm sorry. They are knowledgeable, so they do know that we're doing what we're doing up here. I know downstate, especially in the city, sometimes it seems like that's the New York City is a different state, if not a different country, in terms of, so they are doing something different. I believe they're doing kind of a PEG precipitation, different method. And as far as I know, it's working for them. But I can tell you SUNY is coordinating the analysis.
Great. We got one more last minute question, and how can the public help with the sequencing, or is there anything the public can do to make the sequencing easier or more efficient?
That's a great question. We thought about, especially at ESF, we always think about citizen science, right? How do we get our citizens involved in collecting data? And the thing is, I don't want people going out collecting wastewater samples. So I don't think that's safe. And so, from a sample collection point of view, I don't know if we're ready to go there yet. The sequencing part, there's lots of bioinformatics, and so I maybe a sample collection in terms of might be hard to believe, but we actually like to analyze fecal samples, because that gives us a good gauge on like the relative the abundances of crassphage and SARS-CoV-2 if it's an infected person. So coordinating to send in fecal samples, might be useful. I say that, but now, okay, so now tomorrow, like 1,000 fecal samples are going to arrive at my office door, I'm sure. But so that'll be fun. But that's a great question, and that's something we probably could put a little bit more energy into, thinking about how the public can help with that effort.
All right. Well, I want to say thank you to Professor Green for taking the time. Everybody, do not mail him fecal samples tomorrow. Please do not mail him fecal samples. And I'm going to throw it over back to Mary Beth and the Strathmore speaker. She's going to tell you about some fun things we have coming up. And thank you guys again for joining us.
Thank you.
Thank you, Dr. Green, for joining us here tonight. And thank you for using your expertise to try to get us through this pandemic. Keep in mind that our programs are always on the second Thursday of the month. Our next speaker will be on February 11th, and we will be featuring Suzanne Mettler, a professor of American Institutions and Government at Cornell University. She'll talk about her recently published book, Four Threats: The Recurring Crisis of American Democracy. On March 11th, we will welcome Dr. Sam Gruber, an instructor of art and music history at Syracuse University and founder of Gruber Heritage Global. He will speak about the arts and crafts movement's impact in Syracuse. This talk will be co-sponsored by the Arts and Crafts Society of Central New York. You can find updates on the series at our website, strathmorespeakers.com. Thank you all for joining us tonight, and we hope to see you again in February. Good night.